everyone, welcome to the channel 20th Notebook. I am Dr. Sourabh Kulkarni speaking, and today we will start with quick revision of our subjects for the NEET PG exam or IMI CET exam or FMGA exam. Let's start with the first subject for today, that is biochemistry. We'll have basically a lot of images being covered under this section, also a lot of flowchart based approach, so that you will be able to cover most of the subject in under 30 minutes or probably 30 to 40 minutes. I'll try to cover as much as possible. Beginning with the first topic, that is collagen. So basically how the collagen synthesis happens is in two steps, mainly one in rough endoplasmic reticulum and then in extracellular space. So in rough endoplasmic reticulum there is collagen mRNA translation and production of pre-procollagen, which is then hydroxylated, and then formation of hydroxylysine and hydroxyproline, which is further glycosylated and formed and later three such molecules, glycosylated molecules of hydroxylysine or proline, together form and are linked together and form the alpha chains triple helix structure. So basically this after this is formed, we will have the mature procollagen, which is then exocytosed into extracellular space, where the procollagen is then packaged by the Golgi apparatus, and then it is there is a procollagen peptidase which cleaves the N and C terminals of the collagen, the N and the C terminals, which are then cross-linked. After the cleavage, it forms insoluble tropocollagen, and then cross-linking by lysyl oxidase leads to the formation of mature collagen. Any defective in the following pathway leads to formation of collagen disorders. The first step, where there is hydroxylation, if there is deficit, if there is defect, then we'll see that defect in scurvy, vitamin C, due to deficiency of vitamin C, where there is poor wound healing and a variety of bony deformities which you see, because hydroxylysine and hydroxyproline requires vitamin C. Also in the second important thing was the linkage of three alpha chains, and the defect in COL1A or COL1A1 or COL1A2 leads to the defective linkage of these alpha chains, which is seen in osteogenesis imperfecta, which is also called as a brittle bone disease. Also need to remember that if the procollagen peptidase does not cleave procollagen N and C terminals, then there is a defective disease known as Ehlers-Danlos, where you also know that by the name of sticky man syndrome, or where the patient literally have long hands and very stretchy skins. Along with that, we have the last important step, where there is cross linkage by lysyl oxidase, which requires copper, and this is defective in Menkes disease, as there is defective copper absorption in Menkes disease. This will also lead to defective collagen. So summarizing all this, we have three categories of disorders for collagen, where there is fibrillar disorders, network disorders, and anchoring disorders. In fibrillar, we have 1, 2, 3, and 5 types collagen type disorders, where collagen type 1 is deficient or is defective in osteogenesis imperfecta, as suggested by the gene name of COL1A1 and COL1A2. This is predominantly present in bone and tendon. So remember, bone has collagen 1. Cartilage can also be spelled as cartilage and has collagen 2, which is deficient in achondrogenesis type 2. Whereas Ehlers-Danlos vascular type is deficient in collagen type 3, and Ehlers-Danlos classical type leads to defective collagen type 5. Alport-Goodpasture syndromes, where there is which is present in the basement membrane of lens, cochlea, and glomerulus, has defective collagen type 4. And the vesiculobullous disorder by the name epidermolysis bullosa has defective collagen type 7. So this is our first topic of collagen. Next, moving on to the next topic of lysosomal storage disorders. We all associate lysosomal storage disorders with mainly sphingolipidosis, but also remember mucopolysaccharidosis and mucolipidosis are part of it. Mucolipidosis, you need to remember that There's a disease called as eye cell disease, where the mannose is phosphorylated by Golgi bodies, and it leads to the formation of mannose 6 phosphate. This does not happen in this particular enzyme deficiency called N-acetyl glucosamanyl 1 phosphotransferase, and this leads to the immature production of the proteins, and hence these are then packed by lysosomes, and hence is a lysosomal storage disorder. Then we also have two important mucopolysaccharidosis, Hurlers and Hunters. There are other two, Sanfilippo and Sly, which are not much important for us. As we see, Hurler is the more upstream of the pathway, and Hunter is the more upstream of the pathway, and Hurler comes later on. However, Hurler is more severe and is associated with corneal clouding, and since it is more severe, you can tell that this is a alpha disease because there is deficiency of alpha 1 iduronidase. However, Hunter is less severe, no corneal clouding, and the deficiency is iduronate 1 sulfatase. Okay, moving on to the single lipidosis. Remember that the goal in life is to buy ceramide toiletries basically. So your wash basins and other things which are made up of ceramic, that should be lavish in your life. So basically the goal here is to form ceramide from all these pathways. So basically GM1 converts to GM3 and then converts to glucocerebroside, and then it is converted to ceramide, and sulfatides converted to galactocerebroside and then to ceramide. Sphingomyelin directly converts to ceramide. So what happens here is, if there is deficiency in certain diseases, say Niemann-Pick, it is more closer to ceramide, and then Gaucher's is one step away from getting converted to ceramide. So there is not much correlation between how far the disease is from getting converted to ceramide, but we will use this as a mnemonic based thing, so that we will be easily able to recall them in exam. So what is your main goal? Is to form ceramide from all these substances. So what happens? Tay-Sachs is the most is at least 1, 2, 3 steps away from becoming ceramide. So in Tay-Sachs, it is the vision is very far to becoming ceramide. So the patient has farsightedness. Why is they having farsightedness? Because there is cherry red spots in the macula. So remember, if a patient, if in the clinical vignette there is mention of cherry red spot without any other significant findings, then they are talking about Tay-Sachs. Then also remember the enzyme deficient in Tay-Sachs. That's all. And going to the next important thing, in cherry red, what happens in cherry red in Niemann-Pick? In Niemann-Pick, it is just one step away from becoming ceramide, the sphingomyelin. So the patient has near sight. His vision of becoming ceramide is very near, but still he is not able to become ceramide. Why is that? Because there is a lot of foam in his washroom. So basically on microscopy you will be able to see foamy cells. Why is that? Because of the presence of vacuoles in the cells. Also remember the cherry red spots as illustrated by our story. Then the most important thing in our NEET, PG or NICT is the Gaucher's. So Gaucher's, what is happening with Gaucher's? Gaucher's is just one step away from becoming ceramide again, but still it is unable to reach to become ceramide. That is to have ceramic toiletries or the sinks and wash basins and stuff. Why is it not happening? Because they don't have tissue paper in their washrooms. So what do I mean by this is, on microscopy, the appearance of crumpled tissue. This is one finding you have to remember. Other things you have to remember is the patient will have bone pains. That is because of bone infarcts and other such stuffs. So Gaucher's disease, you need to remember this small story. Other than that, there is one more important disease which you all have to remember, that is metachromatic leukodystrophy. What happens in leukodystrophy is basically white matter disease. So there is dysmyelination or progressive decreased myelination of the white matter. So on MRI, what do we see? We'll be able to see tigroid appearance. Other than that, nothing much is important in metachromatic leukodystrophy. So you just have to remember this small story so that you will be able to recall these 4 metachromatic leukodystrophies. Other than that, we have Hunter's and Hurler's, which are also part of mucopolysaccharidosis and the story which I told before. So other than that, we'll go to the third important topic, which is glycogen storage disease. So how do you remember glycogen storage disorders? We'll have to remember that by 2 important groups, the liver glycogenosis and the muscle glycogenosis. What is happening in muscle glycogenosis is there are 2 groups of disorders where there is muscle energy impairment, where the muscle is unable to provide us with adequate energy because of impairment of, as we know, glycogenesis, gluconeogenesis. And other important thing is the disorders with progressive skeletal myopathy and cardiomyopathy. So if the patient is unable to utilize his muscle energy, which type of disease is he falling into? The 2 important diseases which come under that is McArdle's and Tarrow's, which are type 5 and type 7 respectively. McArdle's, as we know, starts with M and M looks like 5. So remember, McArdle's has type 5 glycogen storage disorders, and there is muscle phosphorylase deficiency. Tarrow's is not that important, where there is deficiency of phosphofructokinase. Other than that, the important thing you have to remember is the type 2 Pompey's. You all have you heard of Battle of Pompey's? So Pompey was a king and he had 2 times the army of Julius Caesar, yet he lost. Why did he lose? Because he had Pompey's disease. Just kidding, this is just for correlation. So what happens in this disease is that the ruler Pompey has got Pompey's disease and hence has progressive skeletal myopathy, and hence he could not defeat Julius Caesar in his war and had to run away. And this is how Caesar began ruling the world. So remember this small story and you will be remembering type 2 Pompey's disease, where there is lysosomal acid alpha glucosidase deficiency. Also in liver glycogenesis, you have to remember there are 2 groups, where the disorders where there is hepatomegaly or the disorders where there is liver cirrhosis. Liver cirrhosis happens only with one enzyme, that is one disease, that is Anderson's. Anderson's starts with A and there is deficiency of enzyme branching enzyme B. It starts with B. So the patient presents with C. So this is your ABC disease. However, there are 3 important diseases in liver glycogenesis which present as hepatomegaly. The first one is von Gierke's. Von, you can correlate it with 1. So what happens here is there is deficiency of glucose 6 phosphatase. However, there is one more important disease, that is Cory's disease, starts with C, where there is deficiency of debranching enzyme which starts with D. So debranching enzyme of liver and or muscle. So there is accumulation of limit dextrans in this particular glycogen storage disorder, and hence is also known as limit dextrinosis or limit dextrinopathy. Next, there is an important one more important disease, that is HERS. HERS disease is type 6 glycogen storage disorder, where there is deficiency of liver phosphorylase. In HERS disease, there is so you have to remember these certain glycogen storage disorders. Other than that, other 2 important not much important are type 9 and type 11, which you don't have to remember. So how will you remember the names? You all know of Corey Anderson, right? He's a cricketer from New Zealand, a batting all-rounder, I would say. So what happened with Corey Anderson? What he could not do? He is like actually very well built cricketer, or you can say fat cricketer. Okay. So what he could not do in his life? He could never run threes. He only took singles and doubles or hit boundaries. So Corey is type 3 disorder. However, what Anderson could also not do is in T20Is he never bowled 4 overs. So Anderson is type 4, Corey is type 3. But one more important thing that you have to remember with Corey Anderson is You all remember that MI versus Rajasthan match where Mumbai Indians chased around 190 in around 14 overs. In that match, Corey Anderson scored some 95 or something not out, and hence he instantly became her favorite cricketer. Who is her in this context? I am talking about Neeta Ambani. Why did he become her favorite? Because he helped Mumbai Indians qualify for the finals, for the playoffs that season, and hence you will always remember that type 6 is hers, because Corey Anderson used to hit sixes, lots of sixes, and in that particular match he hit a lot of sixes and helped them qualify. So this is my short story that that will help you to remember glycogen storage disorders. These are the few important stuff you need to know about glycogen storage disorders. So what are some clinical clues that they will give? As I had told you, Pompe's disease will have cardiomyopathy, as I told, and also presence with normal glucose levels, and there is glycogen accumulation in lysosomes. So other than that, what will you remember with Cory's disease, von Gecke disease and Mcardle's disease? In Cory's disease, there is hepatomegaly also, and there is hypotonia also with ketotic hypoglycemia. What is happening in von Gecke's is there is not ketotic hypoglycemia, there is fasting hypoglycemia. Okay, and there is also lactic acidosis with hyperuricemia. Hepatomegaly will always be present, as they all come under the same category. However, there is one, the Mcardle, which is type 5, is also a muscle glycogenopathy, as I told earlier, with this weakness and fatigue with exercise. So these two will have clues related to weakness, while these two will have clues regarding to hepatomegaly. So remember this small classification and you will be able to answer most of the stuff related to glycogen storage disorders. Okay, moving on to the next part, which is amino acid byproducts. I will not be teaching you in detail the formation of the byproducts through amino acids. I'll just tell you that what is the main vitamin which is being used everywhere here. As soon as you can see that vitamin B6 is essential for formation of every byproduct, almost every byproduct. However, you remember that phenylalanine is the boss here. So is arginine, as they are forming multiple important products. So what does boss, how many letters does boss have? 4. So they all require BH4, that is tetrahydrobiopterin. Okay, for formation of specific products. Rest all are will have vitamin B6 as coenzymes, except one more important stuff, that is formation of NADs from vitamin B2 along with vitamin B6 as coenzymes. So we'll have a small description of the byproducts. Phenylalanine is forming thyroxine through tyrosine. Melanin, your skin pigment, through DOPA. Then we have dopamine also, norepinephrine and epinephrine, all the sympathetic hormones. Then we have tryptophan. What is tryptophan forming? It is forming your NAD plus and melatonin. Remember, this is melatonin, not your melanin. Melatonin, the one which is associated with your circadian rhythm. Okay. Then there is histidine, which is forming histamine. This is easier to remember. Then there is glycine, which is forming your heme and porphyrin. Then there is glutamate, which is forming all the G related, GABA and glutathione. Then the last important thing, arginine. Arginine, as soon as you remember, you have to remember that it forms urea and nitric oxide. Nitric oxide is a vasodilator. Okay, remember this, and remember the vitamin used as coenzymes in forming the following byproducts. Then I'll just have a short detailed metabolic pathway in phenylalanine and tyrosine metabolism, as multiple important disorders are associated with this pathway. So what is happening is, in conversion of phenylalanine to tyrosine, there is involvement of hydroxylation by the enzyme phenylalanine hydroxylase, where BH4 is acting as coenzyme, and if there is deficiency of this particular enzyme, then there is phenylketonuria. Okay, Tyrosine then becomes homogenesic acid, which finally becomes mallyl acetic acid, which is converted to fumarate and then is incorporated into a TCA cycle. So homogenesic acid to methyl acetoacetic acid is mallyl acetoacetic acid is by the enzyme homogenesate oxidase, where there is oxidation, and hence if there is deficiency of this enzyme, then there is alkaptonuria, where the urine becomes black on standing due to the product alkapton. Okay, then in the other alternative cycle, where there is tyrosine getting converted to dopa and then to melanin, dopamine and other sympathetic products, if there is deficiency of tyrosine hydroxylase. Remember, the first two steps here is hydroxylation, where there is attachment of OH group. If OH group is deficient, the enzyme for adding a OH group is deficient in these two pathways, then there is phenylketonuria in the first step and albinism in the second step. So we have three important diseases associated with phenylalanine. What are these three? Your PAR disorders. So we have phenylketonuria, alkaptonuria and albinism. I hope you'll never forget this important cycle too. Next, we have one more short story for remembering ETC, which is completely which is asked usually regularly in at least one exam every year. So what is happening here is there is complex 1, complex 2, complex 3, complex 4 and complex 5, where there is finally formation of ATP. So you have to remember the complex names and the inhibitors at each complexes. So complex 2 does not have any inhibitors, complex 1, 3, 4 and 5 have inhibitors. How will we remember this? Also remember that the ADP to ATP production happens on the mitochondrial matrix side, and this is your inner mitochondrial membrane, and intermembrane space is on the outer side. So we'll just remember one story. When if there is plane hijacking, what does the government negotiate? As you would have seen in other movies, बीवी नहीं, औरतें और बच्चों को पहले छोड़ देना. So what does this mean? They will tell that okay, let the ladies go, let the children go, you get hold of the men. So basically they always sacrifice our men during all such things. So what does complex 1 here stand for? NAD. NAD का मतलब क्या? neonate बच्चूस. So if the bachus are the first ones to get negotiation offer from our government. So how do you associate here bachus? Rotenone. What do bachus do always? They always cry, rote rehte hain. So remember, rotenone is the complex 1 inhibitor. So what will you do next? You will release FADH, that is females. Next negotiation is for females. Females, they will release all the females. Then what happens? Your males become antimycin A. What a complex 3 inhibitors? Antimycin. They will become your antidotes. They are the ones who are going to save us. So basically, antimycin inhibits complex 3. So what happens after that? If the males are unable to save themselves from the hijackers, they will feel that they should better commit suicide. How do they commit suicide? Peaceful suicide is by taking cyanide, which is complex 4 inhibitor, or taking CO, which is carbon monoxide, inhaling carbon monoxide gas, which will help in peaceful death. So after all this, even if the patient, if the hijackers are unable to catch hold of us, what is happening? There is formation of energy by the heroes. That is, ATP is being produced. Our men finally become the heroes. Complex 5 inhibitor, you will have to remember, is oligomycin. This is a small story which can be made to remember your ETC inhibitors and complexes. Okay. And then there is this important sugar disorders which you will have to remember. I have taken this from Mehlman Medical College, medical website, which is for USMLA and is quite a good website. You won't have time for that. So I've usually taken the best resources from the net, and I will help you to learn as much as possible and in a very easy way. Okay. So what is happening in fructose? What happens is this fructose has to be used for glycolysis. So it is getting converted to fructose 1-phosphate and then alternative to fructose 6-phosphate, and then it goes into glycolysis. So in these steps there are two important enzymes, fructokinase, which is the first step, and then aldolase. Okay, if fructokinase is deficient, what is happening? There is essential fructosuria. However, if aldolase is deficient, then there is hereditary fructosuria. Okay, so what is happening in essential fructosuria? The important thing is there is mere presence of reducing sugars in the urine, but the glucose oxidase urine test is negative. Why is that? Because in urine the reducing sugar which is present is fructose, not your glucose. So glucose oxidase urine test is negative, essential fructosuria. Then there is hereditary fructose intolerance. What is happening here? This is happening with a clinical case vignette where a 6-month-old infant is started. The 6-month-old infant is under the presence of a process of weaning, where they are introducing juice, fruit, honey, and other stuff in his diet. So what is happening? As soon as they introduce, the patient is having some symptoms: hepatomegaly, jaundice, hypoglycemia, and all. This is because of the deficiency of aldolase B and non-utilization of fructose in production of energy. Then what happens if the baby is presenting as soon as it is born? It depends on severity with which it is present, less severe or more severe. So less severe may you see only cataract in that patient and some other failure to thrive symptoms or something. So in such cases you have to suspect galactokinase deficiency, where there is only cataract, and this is because of galactitol deposition in the lens. So the patient, as soon as is started with breast milk, presents with this small disorder. Then there is classic galactosemia, where there is Gal1 phosphate uridyl transferase deficiency, where the patient also presents from birth but with cataract and also these important symptoms, which are hepatomegaly, hypoglycemia, jaundice, E. coli sepsis. All these are life-threatening and has worse prognosis than classical galactokinase deficiency. This is some of the sugar disorders which you will have to remember for NEET PG or INI-CET. Okay, then I'll summarize all the stuff which I have taught you and introduce some new steps, new things. So whenever a patient presents with, okay, we will go with a different colored marker, let's say yellow, for some time. So if patient is presenting with high ammonia, what will you do first? You will check its ABG and check if the patient's pH is elevated or not. If the patient's pH is normal, not acidotic, then you have to go with urea cycle defect. High ammonia, then normal pH, then think of urea cycle defect, where there is increased ammonia, decreased burn, and increased pH, especially alkalosis, but it is not acidotic. That you have to remember. Then you have to suspect all the urea cycle disorders and treat as per that. However, if the patient is having acidosis, then you have to think of the organic acidureas. That is the most important thing, where there is bone marrow suppression also. You have to then do a glucose test in other patients, where you can see if the ammonia is normal, then you do glucose test, and then again you check for the same ABG. Then you see if the patient is having normal pH or decreased pH. If the patient is having decreased pH, you have to think of two pathways: glycogen storage disorders. As I have told you earlier, glycogen storage disorder will have what? Decreased pH and decreased glucose in blood, but the glucose is having being stored in other forms like glycogen variants, where in liver or in muscle there is underutilization. Okay, so you have to remember this, and there will always be it need not always have hepatomegaly, as I have told you, but remember these stuffs for the pattern based approach. Also think of one more important thing, that is ketotic hypoglycemia, where one important thing needs to be remembered. There is also as I have told you, decreased glucose and decreased pH, but there is the most important thing: you have to check urine ketone bodies. If there is ketonuria, then it directly goes towards ketotic hyperglycemia and not towards glycogen storage disorder. However, if the patient has a normal pH or raised pH but not acidosis, then think of acid oxidation disorders where there is decreased ketones also and decreased glucose also. And then also think of galactosemia where there is increased urine reducing substance, which is galactose in this case. Okay, so summarizing all this stuff, okay, we'll go with this one. Okay, what is happening here? We have fatty acid oxidation disorders. This is the disorder in metabolism of fat where there is beta oxidation deficiency. So there might be medium chain acyl CoA dehydrogenase deficiency or long chain or very long chain, anything. Okay, the patient is presenting with what? Decreased ketones and decreased glucose. So this is what hypoketotic hypoglycemia. The patient has all these important things: sudden infant death syndrome and Ray's syndrome, lethargy, vomiting. These are important things you need to remember with fatty acid oxidation disorders. Then there is another important thing which I taught you, that is organic acidemias. So organic acidemias has organic acids, not your amino acid related stuff. So organic acid related acidemias are what? Propionic, methylmalonic and isovaleric acidemias. So in this we have HAGMAs. HAGMAs are associated with strokes, coma, death and vomiting. Remember this. However, also remember all this urine related stuff, all the sweet sounding names related to urine: homocystinuria, phenylketonuria, MSID. These all come under amino acidopathies, which are also metabolism defect of proteins where there is no acidosis और hyperammonemia, दोनों नहीं है, अमोनिया भी not raised और acidosis is also not there. Then there is your classical urea cycle disorders where there is ammonia raised. As I have told you here, ammonia is raised. Also, the patient might go into coma, death. It also presents with neonatal lethargy, vomiting and all. So as soon as I see a clinical case vignette where the patient looks like where the neonate looks like having a metabolic disorder, you follow this small flow chart, okay, and you will be able to like solve it in minutes. You will be saving lot of time. Remember this, go through these tables and then you are done with most of the biochemistry related metabolic disorders. Okay, I'll also just quickly brush up one more important stuff. Okay, we haven't covered that. Okay, there is this diagnosis of urea cycle disorders. As because I was talking about metabolic disorders, I'll just quickly complete this. So what are the important things they might ask in urea cycle disorders? You have to remember there is NAGS, NAGS or CPS deficiencies. NAGS is N-acetyl glutamate synthesis and CPS is carbamoyl phosphate synthesis. These are my two picks which are supposedly going to be asked, the author's picks. For all those candidates who have bought our notes, this comes under my author's picks along with the amino acidemias. So remember these two things which can be asked for your current exam, INA or NEET. Okay, so they might ask you NAGS or CPS deficiencies. What is happening here is there is normal urine orotic acid. If they give anything altered with normal urine orotic acid, only think of the first upstream pathways, NAGS or CPS deficiencies. More downstream pathways will have increased urine orotic acids. And remember, there is important thing of raised ASA, that is arginosuccinic acid urea. This arginosuccinic acid is being excreted in urine in two important deficiencies. ASA deficiency, so ASA is deficient, so arginosuccinic acid deficiently will be excreted in urine. उसके downstream pathway कौन सा है? arginase deficiency. तो इन दोनों में if they give ASA elevated along with urine orotic acid elevated, think of these two. Okay, then there is other stuff, but These are the most important pattern based approach for urea cycle disorders. Okay, then I'll go ahead with genetic techniques. तो कैरियोटाइपिंग में क्या करते हैं? कैरियोटाइपिंग में वी विल सी ऐसे कलर नहीं दिखेंगे सब कुछ. यू विल सी सम शॉर्ट आर्म्स एंड लॉन्ग आर्म्स ऑफ ऑल द 23 chromosomes including sex chromosomes. You have to find the defect. Usually they will sometimes mark, जैसे कि यहाँ पे where there is trisomy of what हो रहा है यहाँ पे? Trisomy of 5th chromosome. कौन सा है 5th chromosome trisomy? बताओ. So you have to identify like this on karyotyping. What is wrong with which chromosome? You will have to see. So what is happening in karyotyping is basically if you can see that you can't properly make out unless there is a large a big defect. You can't properly make out if there is a missing short arm or something. Okay, like small gene rearrangement or something can't be made out in this. However, you have to remember that it is a whole genomic approach and can detect balance abnormalities that may predispose that carrier to pregnancy loss. Okay, like bade disorders where there is loss of complete arm or monosomy or something, that can be easily detected on karyotyping and also used in prenatal setting for detection of aneuploidy, which is what I was talking about. However, because of its low resolution, as I told earlier, only large structural rearrangements can be seen. Okay, देखो यहाँ पे there is small arm which is designated with P and long arm which is designated as Q. तो since it is chromosome 6, we either see 6P or 6Q. उसके बाद there are small locus called numbered as 1 2 3 4 5 6 7. उसके बाद सब लोकाई भी है यहाँ पे 1 2 3 4. तो 1.2 ऐसा wherever there is deficit of that particular gene on that chromosome, that will be labeled as such. Then we have another important thing, flow cytometry, where they will tag of antibody with fluorescent label. So this antibody will then go and sit on your antigen on the cell. So cell के ऊपर, if the cell has that particular antigen, then it will get tagged by that fluorescent labeled antibody, and then this will brighten up as soon as you see on fluorescent lamps. So these all flow cyte. उसके बाद they'll separate it as a mixture under flow cytometry. So they allow it to flow under centrifugation method or something. Then they will see. So this technique is adopted in your genetics, in your bone malignancies, hematological malignancies, bone marrow malignancies, ठीक है? Where if say CD5 or CD8, so the cells will initially start from here, you think. So centrifugation okay, there will some of them will go here, some of them will go here. They are ideally CD5, CD8 positive are supposed to go here, because they will get attracted towards these poles. So they will go to this extreme end. बस CD5 पॉजिटिव है और CD8 नेगेटिव है तो यहाँ पे जाएंगे. CD8 नेगेटिव, CD5 नेगेटिव है और CD8 पॉजिटिव है तो यहाँ पे जाएंगे. Remember this small shortcut for solving all those bone malignancy related stuff. Then there is FISH. What is FISH? Fluorescent in situ hybridization. In situ because they just break this gene, the DNA. They denature it so that a single DNA strand, which is going to be hybridized with our probe, which has a nanoparticle that can light up. Okay, so this फिर से फ्लोरेसेंट इसमें देख लेंगे. उसके बाद they will assess if there is any genetic abnormality in that particular patient. So in the particular patient sample. So this is high resolution. Why is this high resolution? Because a particular DNA strand which is coding for a gene is being looked at here. So we can see Williams syndrome जैसे defects and everything. So it looks for specific point mutation in a suspected gene. Okay, that is important thing. But what is it doing in that process? It is looking only at a single gene at one time. It can't look at all the genes at a particular time, because we are giving a probe which is specific to that gene sequence. So you can only see that. So these are your pros for this and cons for this. So it is identifying only single gene at one time and single chromosomal region at one time. Okay, that's all you have to know about that. Then there is PCR. What is PCR? PCR में क्या important steps है? There are 3 important steps: denaturation, annealing, and then there is extension. तो 95 डिग्री सेल्सियस पे denaturation होता है, 45 to 60 डिग्री सेल्सियस annealing, and 72 डिग्री सेल्सियस extension. So what is important to remember is all these are happening at higher temperatures. So you should have enzyme, organic enzyme, which is stable at that temperature. Which enzyme do they use? Stack polymerase. Stack polymerase is produced from Thermus aquaticus. As the name suggests, the microorganism produces the enzyme which is stable at high temperatures also. तो इसके बाद ऐसे cycles, if they repeat it for 50 times, so it will be 2 raised to 50. Each time you will get 2 separate strands. तो 2 raised to 50 क्या होता है? 2 raised to, I remember only like 2 raised to 10 or something, 1024. तो उसको और 40 बार मल्टीप्लाई करना है 2 से 1024 को. So this will be like huge numbers. You get so much quantity of data which can be easily detected. So how is it working is PCR has some types. You have just normal PCR, which I have explained. Then you have RT-PCR, where instead of DNA, reverse transcriptase use करके they will also get data of RNA. Then there is qPCR, which is quantitative PCR. This is real-time PCR. So if they ask you what is the full form of RT-PCR, don't go to real-time PCR. It is reverse transcriptase PCR. There is one more important thing, qRT-PCR, which is quantitative real-time PCR. Okay. Then we have next important topic, that is restriction endonuclease. Okay, moving on to the next one, which is restriction endonuclease. So what does this do is, there's an enzyme, restriction endonuclease, E. coli, for example, extracted from E. coli. So what does it do? It cuts exactly at the palindrome sequences. The palindrome sequences. So what it leads to formation is formation of sticky ends. All these are sticky ends. So these sticky ends are helpful in diagnosis and in gene therapies and other stuff. Okay. Next we'll move on to CGH and microarray reverse hybridization. So what is happening here is the patient's DNA is taken, and then they are being put, the sample DNA is being put into the microarrays, the wells of microarrays, and then the probes, color-coded probes, depending on the initial the patient DNA load, will color them, and depending on the shade of color from darkest to lightest, it depicts the presence of that particular gene in that sample, gene or the protein. Okay, so this can be used for DNAs, RNAs, proteins, and tissue microarrays. Okay, as you can see, the lighter part means that the gene is in sparse amount or is completely absent. Gene in the microarrays, in wells of microarrays in C and E. Okay, however, what is happening in comparative genomic hybridization is that both the normal DNA and the patient DNA are mixed. Say red, this is patient's DNA, which is color coded with red, and now I'll mix it with green. Okay, what is happening now? This was supposed to be, what was supposed to be red plus green mixture has now become a completely different color, which is yellow. So depending on the variable amount, if the patient's DNA has extra amount of the gene, that means there is gain of function mutation, which is being depicted by the well being turned into red. If there is loss of function mutation, that means green is dominating, and hence the well will have green color. Okay, and then the last one, the blotting techniques. You have to remember a small mnemonic: snowdrop. O and O cancel out each other. Southern blot is used for DNA, northern blot is used for RNA, and western blot is used for proteins. Okay, moving on to the second last topic for today, the metabolic fuel usage, what happens? So you think there is a 2 second short burst of energy required for a particular activity, just 2 seconds. What will happen then? All your stored ATP in the cells will get exhausted. The stored ATP is sustainable only for 2 seconds. What will happen after that? You can see the peak rising for creatine phosphate. Your muscles will then work for 10 seconds to provide you additional energy. Then what happens? If you are running, say, a 400-meter race, you will take at least 1 minute of time. So after the initial 10-second burst, where you will exhaust all your stored ATP and creatine phosphate, what will you do further for covering rest 300 meters or say 350 meters or something like that? What will happen? Your anaerobic metabolism comes into play for the first 1 minute. Now say if you are running for more than 2 hours or something, a marathon or something, what will happen? So after the initial 1 minute cycle where anaerobic metabolism also tries to provide you with energy, you will be exhausted and then aerobic metabolism takes over. So remember these metabolic fuel usages during exercise. Also remember that 1 gram of carbohydrate and protein provide 4 kilocalories, whereas 1 gram of fat acid provides 9 kilocalories of energy. Okay, then there is your human chromosome numbers and the following diseases which need to be remembered. Most of them are not that important, and they also the star depicts that there are DNA tests available for these diseases, diagnosing these diseases. But some important things which I would like you to remember among these would be FAP, familial adenomatosis polyposis, is seen in defective chromosome 5. Then I would like you to remember cystic fibrosis, which is seen with chromosome 7. And then you have to remember your sickle cell anemia, chromosome 11, retinoblastoma, chromosome 13, and NF1, which is chromosome 17, also with 5% cases of breast cancer. NF2 is seen with which chromosome? Chromosome 22. Okay, which is not mentioned here. Also remember hemophilia A, which is your X-linked disorder. Okay, these are some of the important things. The rest of them can be ignored. So how will you read a pedigree chart as soon as you give? Don't run away from a pedigree chart. It's very easy and potential 10-second question. That's all. So how will you read that? If you see a female transmitting it to everyone, whereas a male not transmitting to any of them, it's straight away mitochondrial disorders, and you have to remember these examples. However, if you see that both sexes, male and female, in a generation, see one sex or male and one female is involved here, two male and two females are involved here. So all these are seen in what? Autosomal disorders, be it dominant or recessive. Same amount of people are involved. One thing to remember in autosomal dominant is both sexes will transmit to offsprings, and then there are no carriers, and then there is no skip generation. What is happening here? There is skip generations. 3 generations don't have disease. Some of them are carriers. However, suddenly in 4 generation there is appearance of disease. This is happening with autosomal recessive. Then you have to see in X-linked disorders what is happening. Males are more common in X-linked recessive. However, in X-linked dominant, females are likely to be mostly affected compared to males, because males में क्या हो जाता है, males will die as soon as they are having that dominant X gene which is defective. They will die because they have only one copy of that disease, and Y will unable to make balance out that particular defective X gene. So what is happening with X-linked recessive? There is no father to son transmission. That happens with X-linked dominant also. See, fathers are so nice, they never transmit it to their children, but mothers, what do? Even mitochondrial and in other stuff also, they will just transmit it to their daughters or sons. So what is happening here in X-linked recessive? There is skipped generation as seen with autosomal recessive. However, what is happening here? There is no skipped generation. All of the females are usually affected, and all males will transmit it to their daughters. That's all. Okay, now this is all from our biochemistry section. I have not covered certain stuff which you can just read. It might take you another 15 minutes to complete those. One important thing would be all your chondroitin sulfate and all, dermatan sulfate, keratin sulfate and all, which was a PYQ. Then another important thing will be some enzyme defects which I have left out. Don't remember, you don't have to remember the clinical case which case vignettes with which they present. Just remember certain diseases which I have skipped and the enzyme deficiencies in them. Then you'll be covering some more detailed genetics aspect. I have just covered the basics of genetics. You don't have to delve into the enzymes in eukaryotes, prokaryotes and stuff. You just have to see it from a clinical application based point of view, like the genetic techniques which I taught you. Okay, other than that, what will you do next will be some energetics, some pathways, and the most important thing would be vitamins. Okay, just cover vitamins once more. Other than that, you'll be set with biochemistry. That's all they can basically ask. They'll if they ask you around say 20 questions, you'll be able to answer 15 questions by doing these four topics and the topics which I thought you know. So this is how you have to cover it within 40 minutes in the last phase of exam, and you will be done with that. Okay, this is not exactly the same thing which I have used in my notes 20th notebook. I have had much more detailed presentation along with some good mnemonics also in our notes, but this one free video I would have like everyone to watch it so that you'll be more confident. And there are some mnemonics which can't be written in books, and hence I have used it in this particular video. So all those people who are interested to buy our notes, our notes are damn impressive. You will once you see it, you will just fall in love with that. That is why we have had more orders than any other individual notes 20th notebooks. Okay, so you just text us at 8095806118 to buy those notes, or I'll give you a chat in the about description of the video. You just have a chat link to our groups, WhatsApp group. You can just text it there. Other than that, just drop in comment how useful you felt the video was, and other next subject or particular topic you want me to teach you guys. Thank you. That's all I have from today. All the best.